Assalamualaikum and good morning to everyone. My name is Nurul Hassana binti Dawis and I will present my final year project 2 entitled Antimicrobial Activity of Fungal Marasmius cladophyllus UNIMAS MSF. Before that, I would like to thank my supervisor Dr. Simon for the guidance he has given. In this presentation, I will present 7 main sections. The abstract of this study comprises 4 main sections, which are Introduction, Methodology, Result and Discussion, and Conclusion. The introduction focuses on the importance of the discovery of new antibiotic. The methodology introduced 3 main methods used in this project. Result and discussion indicate the ability of Marasmius cladophyllus in performing antimicrobial activity, and the conclusion state the potential metabolite produced by Marasmius cladophyllus UNIMAS MSF. Antibiotic discovery gave a new opportunity to the world in treating infection caused by bacteria. The example of antibiotics are penicillin, polymyxin, and vancomycin. These antibiotics are still in use to this day. However, antibiotic-resistant germs become a concern in the medical world. This is because the same type of antibiotic will not be effective against bacteria that has evolved. Therefore, the experiment was conducted to test the potential antimicrobial activity of endophyte Marasmius cladophyllus. The research question is, does the fungal Marasmius cladophyllus produce metabolite with antimicrobial activity? And the hypothesis is, endophyte Marasmius cladophyllus is a fungi. Thus, it should have the ability to produce antimicrobial activity. Next, there are 2 objectives for this project, which are screening for antimicrobial activity and analyzing MIC. Alright, now we move to literature review. Endophytic fungi are endosymbiont organisms that exist in the tissues of living plants. It has a crucial role in host plant, which is protecting plants by producing a broad range of fungal metabolites. Endophyte Marasmius cladophyllus has limited research papers related to antimicrobial activity. Study from May 2011 related to antimicrobial activity of Marasmius cladophyllus for new sesquiterpene. This indicates the potential of Marasmius species in producing antimicrobial compound. Basically, fungi will produce diversity of secondary metabolites. The secondary metabolite could act as antimicrobial compound. The example of antimicrobial compound from fungus are alkaloid, lactone, and terpenoids. In this project, germs from mouth, hand, and Becker's yeast were used to identify antimicrobial activity of endophyte Marasmius cladophyllus. Usually, most common germs on the skin are E. cloacae, K. pneumonia, and E. coli. Meanwhile, actomyces and lactobacillus are bacteria commonly found in mouth. Lastly, Saccharomyces cerevisiae is a common fungal used in the Becker's yeast. In this research, screening of antimicrobial activity was done by using 3 main methods, which are distribution method, MIC, and MEC. Next, methodology. In this part, I will explain 4 sections. The first one is cultivation of endophyte Marasmius cladophyllus. Second one is extraction of fungal growth. Third, preparation of germs from mouth, hand, and Becker's yeast. And the last one is antimicrobial activity assessed. For your information, the experiment was conducted in hybrid mode, in which laboratory and home material were used according to functional suitability. The cultivation started by inoculating 5 mm of diameter mycelial discs of endophyte Marasmius cladophyllus on petri dish containing MEA. After that, the petri dishes were incubated for 15 days at room temperature. Next, extraction of fungal growth. Initially, 15 plaques of 5 mm fungal mycelium were prepared. After that, in triplicate, 5 plaques of 5 mm fungal mycelium was inoculated onto 100 ml of MEB for 9 days at room temperature. After incubation, the fungal culture was filtered and the clear culture media was dried over the stove with low heat to dry. For preparation of germ from mouth, hand, and Becker's yeast, 1 ml of nutrient broth was prepared and put inside appendix tube. Then clean cotton bud was used for mouth swab and dipped into the 1 ml of nutrient broth. The culture was incubated for 48 hours at room temperature. The steps were repeated for hand swab and Becker's yeast preparation. Next, antimicrobial activity assessed using this diffusion method. Firstly, the suspension of mouth swab, hand swab, and Becker's was spreaded aseptically onto MHA. After that, the paper disc was saturated with the crude extract solution by dipping the paper disc into the extract and placed carefully on the agar plates by using forceps. Then the plates were incubated at room temperature for 24 hours. Size of inhibition was calculated in centimeter. Sample showing larger zone of inhibition was tested for further MIC. Now we move to antimicrobial activity assessed using broth macrodilution. This is done to determine minimal inhibitory concentration. As shown on the slide, this is the DIY version of broth macrodilution by using appendix tube. 1 ml of MHB plus culture and 1 ml of MHB only were dispersed into sterile appendix tube respectively. After that, by using a shearing, 1 ml of Mueller-Hinton broth was dispersed into appendix tube 1 to appendix tube 4 aseptically. Then 5 drops of extract solution, which is approximately 250 microliters, was dispersed into appendix tube 1 only. Then serial dilution was done by transferring 250 microliters from appendix tube 1 to appendix tube 4. Then 250 microliters from appendix tube 4 was discarded before adding 5 drops of Becker's culture into appendix tube 1 to appendix tube 4. The MIC was determined by observing broth cloudiness and bacterial pellet formation at the bottom of the container. For antimicrobial activity assay using minimum bactericidal concentration, this is done after MIC determination of the fungal growth of the endophyte Marasmius cladophyllus. Appendix tube, which show no visible sign of germ growth, was heated on MHA and left for 24 hours at room temperature. This was accomplished by observing for germs growth on pre-incubated and post-incubated agar plates. Next, result and discussion. The first one is cultivation of endophyte Marasmius cladophyllus by using MEA. The growth of endophyte Marasmius cladophyllus was shown on figure 2. On the 5th day, there is contamination on the agar. It is indicated by the black arrow. Contamination problem was able to be solved by subculturing the fungal culture onto first MEA media while taking precaution not to transfer any of the contaminant. Second, cultivation of endophyte Marasmius cladophyllus by using MEB. A total of 3 fungal mycelia were cultivated into MEB for 9 days at room temperature. On the 3rd day of cultivation, bottle A and bottle B become slightly cloudy. This may be due to the contamination while doing the procedure or particle in the broth. The objective of the procedure is to allow the fungal mycelium to produce secondary metabolite. Third, extraction of fungal growth by using distillation method. It is shown that plus minus 0.3 ml of fungal growth was obtained from the extraction. The color of the fungal growth is dark brown with a sticky state. Next, culture preparation from mouth swab, hand swab, and Becker's. In table 1, it can be seen that the culture media was quite cloudy compared to the control media. This indicates the presence of germ growth in the media. Alright, now we move to distribution result. In this procedure, hand sanitizer was used as a control. As you can see, control has shown positive result toward the germ as stated in table 2, especially hand swab result. There is no microbial growth on the MHA. In this research project, some plates do not have zone of inhibition. This may be due to the inability of the compound in the extract to perform antimicrobial activity, or the compound in the extract are insufficient to perform antimicrobial activity. It can be seen on table 3 and 4. Lastly, Becker's. The crude air extract show clear zone of inhibition as can be seen on the table 5. Next, minimum inhibitory concentration. The crude air was tested against Becker's sample for MIC as shown on the table 6. The results show that only appendix tube 1 do not show any changes after compared with control. Thus, the procedure was continued with minimum bactericidal concentration to check whether the media in the appendix tube have no sign of growth from Becker's culture. Next, minimum bactericidal concentration. From the result obtained, all of the plates show growth sign of the Becker's yeast. This means that the fungal growth extract only inhibited yeast growth and not able to kill the yeast cell. In conclusion, The result demonstrated that crude extract from the fungal Marasmius cladophyllus UNIMAS MSZ contain antifungal activity. It can be used as an attractive source of pharmaceutical. Next, these are my list of reference. You can further check for more information. Before I end my presentation, I would like to thank UNIMAS, my supervisor Dr. Simon, my family, and my friends for the support and guidance they have given. Thank you very much for your attention.